Nature of Science article called Unlocking the Secrets of Cells. It takes courage and dedication to follow your dreams. Lydia Villa-Komarov learned that early in her career. She comes from a family of courageous Mexican-American women. Her mother and both grandmothers were strong role models for her. Their support, as well as her father's, encouraged her to pursue a career in science. As a molecular biologist, Dr. Villa-Komarov studies the role of proteins in the growth and development of living things. In 1976, Lydia was part of a team conducting genetic engineering, a technique by which scientists transfer genes from one organism into another. Today, scientists use this technique to produce medicines, to treat diseases, and to improve crops. In the 1970s, genetic engineering was a new idea. It was feared by many people who thought it might have harmful results. In fact, the city where Lydia worked banned genetic engineering. To continue her research, Lydia was forced to move her lab to another state. She spent a year away from many of her colleagues and friends. It was a frustrating and lonely time, she recalls. Here's just a little bit about her. She is the vice president for research at Northwestern University in Chicago, Illinois. She earned her PhD in cell biology at MIT, and she's an avid skier and photographer, apparently. She likes to read. Kind of cool. She went to MIT, which is right here in Boston, well, technically Cambridge, and Northwestern University in Chicago. If you know Mr. Smith on the Aquarius team, that's actually where he went to school as well. Kind of cool. All right, anyway. But her hard work paid off. The ban was lifted, and soon after, Lydia helped discover a method for making insulin. Insulin is used to treat people who suffer from diabetes. Discovering a way to make insulin launched a new industry, biotechnology. It marked a personal triumph for Lydia. Scientifically, that was the most exciting time of my life, she says. There were any number of reasons to think we couldn't make insulin, but we planned it, we tried it, and no experiment before or since has worked so smoothly. Many secrets of the human cell remain to be unlocked. Lydia hopes to provide some of the keys. Little Q&A session with Dr. Villa-Komarov. How did you become interested in science? My Mexican grandmother was very interested in natural history, plants in particular. She had books we used to look at with beautiful color pictures of plants. What really sparked my interest was just following her around, learning about the plants in our garden, and going out collecting wild spinach with her. What made you choose a career in biology? I had an incredibly exciting developmental biology course in college. One time, we camped out in the lab for 36 hours so we could watch frogs develop. Normally you study that in pictures and textbooks, but we were seeing it happen in real life. It was very exciting. What does a molecular biologist do? We study development at the most basic level. What goes on within a cell? Think of the cell as a house with many different parts, the foundation, the walls, the roof, and lots of bricks and wood and wiring. I'm interested in finding out how that structure gets built. Let's take a look at this picture for a second. The gene for human insulin is isolated from its chromosome inside a cell. This is how they made the insulin, okay? Then number 2, plasmids, which are small circular rings of DNA, are removed from bacterial cells. The insulin gene is inserted into the bacterial plasmid. Let's go down. Then plasmids with the insulin gene are taken up by the bacterial cells. The gene directs the cell to produce insulin. So basically, the DNA gets put into a bacterial cell, and the bacterial cell produces more insulin. And then at the end, the insulin is created and used to treat people with diabetes. It's really cool. Okay, next question. Is there a plan or blueprint for building a cell? All the instructions are in the DNA. That is the material in the nucleus of a cell that carries the hereditary information that determines traits, such as your skin and hair color. The machinery, which is made up of proteins, comes in and reads bits of that information, which are called genes. Then DNA is copied into RNA, a message that travels out to the part of the cell where all the building activity goes on. Other proteins read it and start to produce the materials that the cell needs to work. Are proteins the tools or the structure of a cell? Actually, they're both. proteins are the building blocks of cells, like the bricks and mortar in a house. But they're also the machinery that builds cells, like hammers and drills. Proteins make up the cell and build the cell. What other information does DNA contain? It contains coding instructions to make sure that the right information gets used at the right time. If you were building a house, you couldn't put up the roof before you had walls. In building a human, a certain amount of the head needs to be in place, for example, before you can make eyes. So it's very important for a cell to know what information to use and when to use it. How do cells know when to start and stop building? It's still not clear how that process is coordinated. There are certain genes that we understand very well. We know what signals they send the cell to say it's time to become a heart or a liver. But how does the cell know when to use that information and when to stop? Those are some of the big questions that we're trying to answer. What was the significance of the technique you developed to produce insulin? People with diabetes used to be treated with pig insulin, but pig insulin is in short supply and therefore expensive. The work we did showed how to make a lot of insulin very cheaply by growing it in bacteria, and it's human insulin. How did you trick the cell into making insulin? We were able to isolate the gene with instructions for making insulin. We then inserted it into bacteria. Basically, we tricked the bacteria into thinking the gene was one of its own pieces of DNA. The bacteria then made the appropriate RNA, which was then translated into insulin. Why didn't you give up when your research was banned? We were doing very important work. To quit would have been to admit that the ban was right. We knew that others were doing the same research. You can't win a race if you quit. Were you ever discouraged? There were times when I was discouraged, but I think that is true of anyone doing something where there is no guarantee of success. I think I've always approached a project with the idea that I have to give it my best shot. What advice would you give a person planning a science career? You can't be entirely sure what you'll be able to do. The world is changing too fast. The important thing is to find something you like and learn it very well. If you follow what you like, it may be different in 10 years, but will be a logical extension of your own interests. All right, let's move into the textbook sections. This one's on Mendel's work. All right, remember taking a look over here at the reading preview is very important. So when you ever get reading like this on your own, you should make sure that you're taking a look at that. So you want to make sure you're reading the key concepts, which are what were the results of Mendel's experiments or crosses, something you want to keep in mind as you listen and as you read. And then also what controls the inheritance of traits in organisms. These are the key terms here: heredity, trait, genetics, fertilization, purebred, gene, alleles, dominant allele, recessive allele, and hybrid. I actually really believe in outlining as a form of note-taking, so if you have any interest in taking a look at learning how to outline better, definitely feel free to stop by my extra help. I'd be happy to help you. Even if you're not on my team. So basically you just put the main point as a number 1, and then you put all of like the bullets underneath. You could just do bullets, you can do the letters, and these would just basically be like supporting points that help explain the main point here. So what I often find is that the main point is in bold, and then the supporting points are in the paragraphs underneath that bold text. The main ideas from each paragraph. All right, so let's get going. In the mid-19th century, a priest named Gregor Mendel tended a garden in a Central European monastery. Mendel's experiments in that peaceful garden would one day revolutionize the study of heredity. Heredity is the passing of physical characteristics from parents to offspring. Mendel wondered why different pea plants had different characteristics. Some pea plants grew tall, while others were short. Some plants produced green seeds, while others had yellow seeds. Each different form of a characteristic, such as stem height or seed color, is called a trait. Mendel observed that the pea plants' traits were often similar to those of their parents. Sometimes, however, the plants had different traits from those of their parents. Mendel experimented with thousands of pea plants to understand the process of heredity. Today, Mendel's discoveries form the foundation of genetics, the scientific study of heredity. I know I've said in the past that the beginning part of a reading typically is just fluff, but this one really dives right in and has 3 important things that you need to write down, which you can tell are important because they're in bold. All right, Mendel's experiments. Figure 1 shows a pea plant's flower. That's hard to say. Let's take a look really quickly. Here's a pea plant, okay, and what he did to prevent self-pollination, Mendel removed the pollen-producing structures from the flower, the pink flower in this case. He used a brush to remove the pollen from a white flower on another plant, and then he brushed the pollen into the pink flower. So he was cross-breeding the pink and the white plants. And then the egg cells in the pink flower were then fertilized by sperm from the white flower. After a time, peas formed in the pod. So basically, they're just showing you how he very, very meticulously, can you imagine, he had to do this on so many plants, went in and cut out all of the little stems inside that had pollen, then took the pollen from one and brushed it into the plant of another. So, okay, here we go. Let's go back here. The flower's petals surround the pistil and the stamens. The pistil produces female sex cells, or eggs. The stamens produce pollen, which contains the male sex cells, or sperm. A new organism begins to form when egg and sperm join in the process called fertilization. So even in flowers, the same things, sperm and eggs, are the words that we use to talk about the sex cells. Before fertilization can happen in pea plants, pollen must reach the pistil of a pea flower. This process is called pollination. Pea plants are usually self-pollinating. In self-pollination, pollen from a flower lands on the pistil of the same flower. So it falls from here down into the plant and fertilizes itself, usually. But what he did was cut it out so that didn't happen. Mendel developed a method by which he cross-pollinated, or crossed pea plants. To cross 2 plants, he removed pollen from a flower on one plant. Then he brushed the pollen onto the flower of a second plant. Crossing pea plants. Suppose you wanted to study the inheritance of traits in pea plants. What could you do? Mendel decided to cross plants with contrasting traits, for example, tall plants and short plants. He started his experiments with purebred plants. A purebred organism is the offspring of many generations that have the same trait. For example, purebred short pea plants always come from short parent plants. All right, the F1 offspring. In one experiment, Mendel crossed purebred tall plants with purebred short plants. Scientists today call these parent plants the parental generation, or the P generation. So the first set that you're starting with is the P generation. The offspring from this cross are the first filial generation, or the F1 generation. The word filial comes from filia and filius, the Latin words for daughter and son. In Figure 2, notice that all of the offspring in the F1 generation were tall. Even though one of the parent plants was short, none of the offspring were short. The shortness trait seemed to disappear. So let's take a look at the picture over here. He crossed a tall plant and a short plant in the P generation, the first set that you're looking at. Then in the F1 generation, both the offspring were tall. So he crossed them together. And then in the F2 generation, he got 3 tall plants and 1 short plant. When the plants in the F1 generation were full grown, Mendel allowed them to self-pollinate. Surprisingly, the plants in the F2 generation were a mix of tall and short plants. The shortness trait had reappeared, even though none of the F1 parent plants were short. Mendel counted the tall and short plants. About 3/4 of the plants were tall, while 1/4 were short. So these two, so they pollinated themselves, and then that's how they got to the 3 tall, 1 short. Experiments with other traits. Mendel also crossed pea plants with other contrasting traits. Compare the 2 forms of each trait in Figure 3. In all of Mendel's crosses, only 1 form of the trait appeared in the F1 generation. However, in the F2 generation, the lost form of the trait always reappeared in about 1/4 of the plants, and about 1/4 of the plants. So, let's take a look at Figure 3. I'm having trouble figuring out what figure 3 is. It must be this. So he figured out, let's see, is there anything else down here? No. So he figured out by doing his crosses what was the dominant allele and what was the recessive allele. So, like we just saw with the tall and short, right? When the P generation was bred together, crossbred, it produced just tall plants in the F1 generation. The first set of offspring is the F1 generation. But then when those self-pollinated and created more plants, there were 3 tall and 1 short. That means that probably that 1 short is the recessive allele. So here are some of the other stuff he was looking at. So wrinkled, whether it was green or yellow. There's a coating on the seeds, whether it was gray or white, smooth or pinched. Pockets on the outside. You've probably seen those different types of pockets in peas that you've seen. All right. Let's keep reading. Dominant and recessive alleles. Mendel reached several conclusions on the basis of his experimental results. He reasoned that individual factors or sets of genetic information must control the inheritance of traits in peas. The factors that control each trait exist in pairs. The female parent contributes one factor, while the male parent contributes the other factor. Finally, one factor in a pair can mask or hide the other factor. The tallness factor, for example, masked the shortness factor. Genes and alleles. Today, scientists use the word gene for the factors that control a trait. Alleles are the different forms of a gene. The gene that controls stem height in peas, for example, has 1 allele for tall stems and 1 allele for short stems. Each pea plant inherits 2 alleles from its parents, 1 allele from the egg and the other from the sperm. A pea plant may inherit 2 alleles for tall stems, 2 alleles for short stems, or 1 of each. An organism's traits are controlled by the alleles it inherits from its parents. Some alleles are dominant, while other alleles are recessive. A dominant allele is one whose trait always shows up in the organism when the allele is present. A recessive allele, on the other hand, is hidden whenever the dominant allele is present. A trait controlled by a recessive allele will only show up if the organism does not have the dominant allele. Figure 3 shows dominant and recessive alleles in Mendel's crosses. That's what we were just looking at. In pea plants, the allele for tall stems is dominant over the allele for short stems. Pea plants with 1 allele for tall stems and 1 allele for short stems will be tall. The allele for tall stems masks the allele for short stems. Only pea plants that inherit 2 recessive alleles for short stems will be short. Alleles in Mendel's crosses. In Mendel's cross for stem height, the purebred tall plants in the pea generation had 2 alleles for tall stems. The purebred short plants had 2 alleles for short stems. The F1 plants each inherited an allele for tall stems from the tall parent and an allele for short stems from the short parent. Therefore, each F1 plant had 1 allele for tall stems and 1 for short stems. The F1 plants are called hybrids. A hybrid organism has 2 different alleles for a trait. All the F1 plants are tall because the dominant allele for tall stems masks the recessive allele for short stems. When Mendel crossed the F1 plants, some of the offspring in the F2 generation inherited 2 dominant alleles for tall stems. These plants were tall. Other F2 plants inherited 1 dominant allele for tall stems and 1 recessive allele for short stems. These plants were also tall. The rest of the F2 plants inherited 2 recessive allele for short stems. These plants were short. Symbols for alleles. Geneticists use letters to represent alleles. A dominant allele is represented by a capital letter. For example, the allele for tall stems is represented by a capital T. A recessive allele is represented by the lowercase version of that letter. So the allele for short stems would be represented by a lowercase t. When a plant inherits 2 dominant alleles for tall stems, its alleles are written as capital T, capital T. When a plant inherits 2 recessive alleles for short stems, its alleles are written as lowercase t, lowercase t. When a plant inherits 1 allele for tall stems and 1 allele for short stems, its alleles are written as capital T, lowercase t. Let's do this one actually. Figure 4, black fur and white fur. In rabbits, the allele for black fur is dominant over the allele for white fur. What combination of alleles must the white rabbit have? Remember, black is dominant, white is recessive. Hopefully, you realize that the rabbit must have 2 recessive alleles. So if we're talking about black being dominant, we'd say 2 lowercase b's. I just chose B because black is the dominant trait, B for black. So the white rabbit would have 2 lowercase b's as his alleles. All right, significance of Mendel's contribution. Mendel's discovery of genes and alleles eventually changed scientists' ideas about heredity. Before Mendel, most people thought that the traits of an individual organism were simply a blend of their parents' characteristics. According to this idea, if a tall plant and a short plant were crossed, the offspring would all have medium height. However, when Mendel crossed purebred tall and purebred short pea plants, the offspring were all tall. Mendel's experiments demonstrated that parents' traits do not simply blend in the offspring. Instead, traits are determined by individual separate alleles inherited from each parent. Some of these alleles, such as the allele for short height in pea plants, are recessive. If a trait is determined by a recessive allele, the trait can seem to disappear in the offspring. Unfortunately, the importance of Mendel's discovery was not recognized during his lifetime. Then, in 1900, 3 different scientists rediscovered Mendel's work. These scientists quickly recognized the importance of Mendel's ideas. Because of his work, Mendel is often called the father of genetics. All right, keep on rolling. Here we are, phenotypes and genotypes. 2 useful terms that geneticists use are phenotype and genotype. An organism's phenotype is its physical appearance, or visible traits. An organism's genotype is its genetic makeup, or allele combinations. To understand the difference between phenotype and genotype, look at Figure 9, over here on the left. The allele for smooth pea pods, capital S, is dominant over the allele for pinched pea pods, lowercase s. All of the plants with at least one dominant allele have the same phenotype. They all produce smooth pods. However, the plants can have 2 different genotypes, 2 capital S's or capital S lowercase s. If you were to look at the plants with smooth pods, you would not be able to tell the difference between those with the 2 capital S genotype and those with the capital S lowercase s genotype. The plants with the pinched pods, on the other hand, would all have the same phenotype, pinched pods, as well as the same genotype, 2 lowercase s's. Geneticists use 2 additional terms to describe an organism's genotype. An organism that has 2 identical alleles for a trait is said to be homozygous for that trait. A smooth pod plant that has the alleles capital S, and a pinched pod plant with the alleles lowercase s are both homozygous. An organism that has 2 different alleles for a trait is heterozygous for that trait. A smooth pod plant with the alleles capital S, lowercase s is heterozygous. Mendel used the term hybrid to describe heterozygous pea plants. Codominance. For all of the traits that Mendel studied, one allele was dominant while the other was recessive. This is not always the case. For some alleles, an inheritance pattern called codominance exists. In codominance, the alleles are neither dominant nor recessive. As a result, both alleles are expressed in the offspring. Look at Figure 10 on the right here with the chickens. Mendel's principle of dominant and recessive alleles does not explain why the heterozygous chickens have both black and white feathers. The alleles for feather color are codominant, neither dominant nor recessive. As you can see, neither allele is masked in the heterozygous chickens. Notice also that the codominant alleles are written as capital letters with superscripts. So capital F with a B superscript for black feathers, and capital F with a W superscript for white feathers. As the Punnett square shows, heterozygous chickens have the capital FB and capital FW allele combination. So you don't have to worry about being able to do this kind of Punnett square, but just kind of understand that codominance exists, where sometimes neither one is dominant over the other. And that's how you can sort of get mixtures. All right, that's all we have to talk about. So if you have any questions, definitely come by Extra Help. Hopefully you found this helpful.